this time we are going to interpolate and discuss the reaction or immunofluorescence reaction on antinuclear antibody testing. So it is called IIF-ANA, which is indirect immunofluorescence antinuclear antibody testing. So this is actually even the best methods to detect antinuclear antibodies, of course, but there is also another technique we can use, which is enzyme-linked immunosolvent assay. So demonstrable antibodies, including antibodies against nuclear components, antibodies against cell surface, and even antibodies against cytoplasmic antigens of some cells, including lymphocytes in their classes, polymorphonuclear cells, more specifically, of course, neutrophils, lymphocytic leukocytes, erythrocytes, platelets, and also neuronal cells of the central nervous system, and also, of course, antibodies against immunoglobulin G, so we call them anti-IgG. So at present time, immunofluorescence is the most widely used technique for antinuclear antibody screening, and assays are helpful in assessing renal disease associated with systemic lupus erythematosus are the antibodies against double-stranded DNA, antibody against levels of immunoglobulin, complement 3, C3 and C4, and also cryoglobulin's antibodies. So indirect immunofluorescence testing for antinuclear antibody is based on fluorescence conjugate antiglobulins. So what's the meaning of fluorescence conjugate immunoglobulin antiglobulins? So those are anti-human antibodies that are fluorescence conjugated, which means, of course, we are using fluorescence in order to detect those that reaction between those anti-human antibodies and human antibodies, because what we are detecting here is not any antigen, but it is a human antibody, which of course in form of autoantibody. So we are using, of course, anti-human antibodies that are labeled or that are conjugated with fluorescence. So in one assay, the serum specimen is derived into a well on a microscope slides that contains fixed mouse or rat liver or kidney substrates. So we are using a mouse or rat liver substrates, or sometimes kidney substrates, or cell cultured fibroblasts. So those are substrates, so which means they exhibit antigens. So those antigens are substrates for human antibodies. So in serum or in serum specimen, we expect human antibodies, of course, those autoantibodies that are going to react with substrate antigens, of course, nuclear antigens on the substrates. And if those antibodies are present in serum of the patient, of course, the unlabeled antibodies will attach to the nuclei of the cell in the substrates. So this is what is going to happen, of course, because we are going to use our substrates, which is kidney substrates or liver substrate cells, or cell cultured fibroblast. So those antibodies in serum we are going to detect are autoantibodies, of course, in any autoimmune disease. And if those antibodies are present, because they are not labeled, they are not conjugated with the fluorescence, they will attach to the nuclei of those cells in the substrate. So that's what will happen. So later, after the substrate is washed in buffer, the slide is then incubated with the second or secondary antibodies that are fluorescence labeled. Those are anti-human globulins or anti-human antibodies, and those antibodies, they will attach to the substrate bound antibodies. So this means those antibodies which are human anti-human antibodies, they will attach to the substrate bound antibodies. So those substrate bound antibodies are human autoantibodies that we are trying to detect. So that then fluorescence will be seen microscopically using ultraviolet light. So this is, of course, what will happen, and the slide should be examined as soon as possible, or if not possible, then the slide can be stored in dark at 4 degrees, and this can last for 48 hours before read. So, but several other different patterns of fluorescence reactivity are depending on which antinuclear antibodies have reacted with the whole nucleus or with nuclear components such as nuclear proteins, DNA, or histones. So it depends on which antibodies, on which antigens in the nucleus or the whole nuclear have reacted with antibodies, of course, of the patient. Because we are just using antigens, substrates, as just a substrate, and we are detecting serum antibodies of these patients. So patterns are described as being diffuse, as being homogeneous, as being peripheral, as being speckled, or nucleolar fluorescence. So it depends on the pattern of those autoantibodies. Depending on the pattern, then you can be able to interpret and also try to identify which antibody might be involved in this reaction. So the first pattern that we identify here, it is what we call nuclear rim, or sometimes is also called peripheral pattern. Peripheral pattern is associated with antibody to native DNA and deoxyribonuclear protein, and bear a correlation with the systemic lupus erythematosus, SLE activity, and lupus nephritis. So this is what you call rim or peripheral pattern. Peripheral pattern here is associated with double-stranded antibodies against double-stranded DNA, and also antibodies against deoxyribonuclear protein. So deoxyribonuclear protein as well as antibodies against native DNA. So as you remember, native DNA is also what we call a double-stranded DNA. So when you see this nuclear rim or peripheral pattern, that is suggestive of systemic lupus erythematosus, because that is where we find antibody against double-strand DNA, but not really specific. It is just correlation, but not really so specific, because even antibody against double-stranded DNA can also be seen in other conditions. But of course, always systemic lupus, we must find antibodies against double-stranded DNA. So the second pattern is called homogeneous pattern, which is also diffuse pattern. This is also suggestive of systemic lupus erythematosus or another connective tissue disorder. So another one, which is called speckled pattern, is found in many diseases, including systemic lupus erythematosus. And nucleolar pattern is normally associated with progressive systemic sclerosis or Sjogren syndrome. So I hope you remember these disorders: PSS, what you call scleroderma, and Sjogren syndrome. So the normal person is expected to have a negative reaction. No green or gold fluorescence is observed. If patient does not have any autoantibodies in their serum, so then it means no green color is developed. Of course, always green color means there is fluorescence of detected autoantibodies in serum of the patient. So the degree of positive fluorescence may be semi-quantitated on a scale from 1 to 4 plus, and positive sample give a green-gold fluorescence, which is of course a characteristic of homogeneous, peripheral, speckled, or nucleolar reaction. So before we interpret every pattern or every pattern of reaction, I just want to remind you that it is very important for you to go through this topic very carefully, because most of the questions in your immunology, ACP immunology, most of the questions you have to expect questions from autoimmune disorders, and they like to know how are you able to interpret reactions in the antinuclear antibody testing. So that's very, very important. And for that information, it was very difficult for me to get this content. Very difficult, because I used to see questions. Most questions, ACP questions, they focus on interpretation of patterns in indirect fluorescence antibody testing, but it was very difficult for me to find a book that I can get this content. I think I had the first contents. I think my very first groups, we had a very bad content on this, because it was very hard for me to find the content. So, but I tried to get for them some content that I was able to afford. This one I got it later, a bit later, so it was not easy. So, but as now we have everything, so I hope now you have to take your time and go through them clearly and try to understand, because they are very clear. And you can be able to understand and be able to interpret any reaction or any pattern that you observe. So because antinuclear antibodies react with the whole nucleus or just nuclear components, for example, a protein of the nuclei, DNA or histone, so the reaction pattern reflects the distribution of antigen in the nuclei. So that's very important. So we are trying to explain that the reaction that you observe is consistent with the distribution of antigen in the nuclei or in the nucleus. So when the antigen are distributed along the rim of the nucleus, that's when you are able to see what we call peripheral reaction. If the antigen are just located in the center of the nucleus, you are going to see another reaction, which is for example, nucleolar reaction. Or sometimes antigen are scattered in the nucleus. So sometimes you are going to see speckled reaction, because speckled reaction shows the scattered reaction of the immunofluorescence. So the following are targeted. In the fluorescence testing. So some of them can target double-stranded DNA, others chromatin. For example, anti-Smith, sometimes smooth antibodies, ribonucleoproteins, soluble substance A, soluble substance B, SCARE70, and Centromere, JO1 cyclic citronated peptides. This is also another important protein. So we will see this one in rheumatoid arthritis, cyclic citronated peptides, the CPP. So it depends on which antigen is detected in the nucleus, so which is also consistent or equivalent with which antibodies these patients exhibit in the serum. So the first is, as described before, is diffused or homogeneous pattern antinuclear antibody reaction. So the diffuse or homogeneous is characterized by is a characteristic of anti-DNA nucleoprotein antibody, which is antibodies to native DNA or antibodies to double-stranded, antibodies against single-stranded DNA, antibody against deoxyribonucleoprotein, as well as antibodies against histones. So when you detect antibodies against those antigens in the nucleus, in fact, we are likely to find homogeneous pattern. So even though vacuoles may be seen sometimes, the whole nucleus fluoresces evenly, because as you can see, DNA is almost all over the nucleus, and even depending on the second protein that is all over it is deoxyribonucleoprotein and histones. These are proteins. So they are detected all over the nucleus, in the whole nucleus. So you will see nucleus fluorescing evenly, the whole nucleus will fluoresce. So this pattern is seen in rheumatoid arthritis, rheumatoid disorders, and high titer of homogeneous, of course, is a suggestive of systemic lupus erythematosus, but low titers may be found even in some other syndrome, for example, Sjogren syndrome, mixed connective tissue disease, as well as rheumatoid arthritis. So, but high homogeneous reaction antinuclear antibodies is a suggestive of systemic lupus erythematosus. So this is how it looks like. You can see that homogeneous normally it shows homogeneous fluorescing of the nucleus. Sometimes you will be able to observe nucleus, which means sometimes you will see some spots where this fluorescing is not really homogeneous or is not really evenly distributed. But most of the cell, for example, like this one, as you can see, like this one, this nucleus, and even this nucleus, doesn't show any nucleolus. It shows even distribution of the fluorescence. So that's what you call homogeneous. And as you can see, it suggests that the antibodies that are detected are those antibodies against DS, double-stranded DNA, single-stranded DNA, and deoxyribonucleoprotein, as well as histone antibodies. So that is a suggestive of systemic lupus, even if it is not really specific for systemic lupus, but high dose or high fluorescence, it suggests, of course, systemic lupus erythematosus. The second reaction, it is what you call a peripheral pattern. So the peripheral pattern is also another pattern, and it is also called a marginal pattern or rim pattern, and this results from antibodies against against DNA, which means, of course, it is either not that it is end. Here is not end, but it is either native DNA or double-stranded, of course, or deoxyribonucleoprotein. So if this peripheral pattern is observed, it means those antibodies detected are against double-stranded DNA or deoxyribonucleoprotein, but not other types of antigen in the nucleus. So the central protein of the nucleus is only lightly stained or not stained at all, but the nuclear margin fluorescent strongly and appear to extend to the cytoplasm. So this pattern is associated with systemic lupus in active stage of the disease and sometimes in Sjogren syndrome. So that is the thing. So now peripheral pattern, normally it is associated with antibodies against double-stranded DNA. That is what I would like to remind you. It is antibodies against double-stranded DNA, and it shows the rim or marginal pattern, and this always suggests the presence of a systemic lupus erythematosus in its active stage, or sometimes with Sjogren syndrome. So you will see that that fluorescence will even extend a bit to the cytoplasm of the cell. So you can see here, it shows, it says that in the center of the nucleus, sometimes you can also see some fluorescence, but the fluorescence is accumulating at the rim of the nucleus, and also sometimes extending to the cytoplasm. So here you see the black color is the cytoplasm of the cell, because cytoplasm is not fluorescing at all. So, but you can see that some of the feathers are extending to the cytoplasm of the cell. So that is always a confirmation of peripheral pattern, which of course always suggests anti-double-stranded DNA, which is a suggestive of the systemic lupus and sometimes Sjogren syndrome. The third pattern is called speckled, and speckled pattern occurs in the presence of antibodies to any extractable nuclear antigen apart from DNA or histone. So that is also very important. It is any pattern which detects antibodies to any extractable nuclear antigen. So what is the meaning of extractable? This is like we can call it just any soluble antigen, not soluble in plasma, but soluble within the nucleus. So like it is extractable. Yeah, I hope that one is clear. It is extractable antigen in the nucleus. So except DNA or histone, so the antibodies detected are they are detected against the saline extractable or directed against the saline extractable nuclear antigen, anti-ribonucleoprotein, or anti-Smith antigens. So Smith antigen are also found in the plasma, in the nucleus, and they are called extractable antigens. So those antibodies against Smith, they are seen associated with speckled patterns. So grainy pattern will be seen, and numerous round dots of the nuclear fluorescence without staining of the nucleoli, and is seen in the this type of the patterns. Antibody to Smith antigen have been shown to be specific for patients with the systemic lupus erythematosus. You remember that one. Antibodies against Smith antigens, you know what we call anti-Smith antibodies, are normally associated with the systemic lupus erythematosus. So anti-ribonucleoprotein has been found in many rheumatoid disease or rheumatic diseases, for example, systemic lupus, rheumatoid arthritis, Sjögren's syndrome, PSS, or mixed connective tissue disease, or MCTDs, and dermatomyositis. We have discussed all these conditions, right? In that case now, nobody will judge me, right? We have discussed all these conditions: PSS, MCTDs, and also dermatomyositis, as well as systemic lupus. But we have not yet discussed rheumatoid arthritis, but I will try to see the way you can discuss it later. All right, so, but those are the antibodies against ribonucleoprotein. But antibodies, of course, against Smith are specific, very highly specific for systemic lupus. So whenever I find speckled reaction, it is not that it is also specific for systemic lupus, but it is also suspected because speckled reaction can be associated with antibody against Smith or antibody against ribonucleoprotein, which is also found in other conditions like SLE, RIA, Sjögren, PSS, MCTD, as well as dermatomyositis. So this is how we can interpret as speckled reaction. You can see it is a difference from homogeneous, because you would see it is a grainy. It is grainy in appearance. You see some grains there because it is not staining everything here. It shows grainy or coarse in appearance. You cannot compare it with homogeneous, because homogeneous is even fluorescing of the nucleus. The fourth pattern is called nucleoli. Nucleolar pattern, it reflects antibodies against nucleoli ribonucleoprotein, ribonuclear RNA, and which is, of course, ribonucleoprotein 46S. So a few round, smooth nucleoli that vary in size will fluoresce when examined under UV light. So those are a few round, smooth nucleoli that will be fluorescing. Just a few smooth nucleoli within the nucleus. So, but that is what we call it nucleoli pattern. It is just a few round dots in the nucleoli of the nucleus. So they will be fluorescing under UV light. So nuclear pattern is present in 50% of patients with scleroderma, which is, of course, progressive systemic sclerosis, Sjögren's syndrome, and SLE. So some of them exhibit nucleoli pattern. So this pattern can also be observed in undiagnosed type of Raynaud's phenomena. This is also found. But nucleoli antibodies, those antibodies are detected against nucleoli ribonucleic acid, which is ribose nucleic acid, or these subunits of RNP or ribonucleoprotein. So then that's how they look like. You can see that's how they look like. They are just like very small nucleoli fluorescing dots. It is not the same as speckled, because speckled is going to stain almost the entire nucleus, but with the grainy appearing staining, right? But in the nucleoli, you can see the difference is that in the nucleoli you can see only like small dots, like just one dot per nucleus. It can be like one dot per nucleus, or it can even be like 2 dots per nucleus. Like for example here, you can see that there are 2 dots, 3 dots per nucleus, 1 dot, 2 dots per nucleus. So it is not like speckled, because speckled is like the entire nucleus is staining, but there are some dots which are nucleoli. So nucleoli are not stained in speckled. So that's why speckled, it shows almost homogeneous, but with some dots which are the nucleoli. So they are not stained in the speckled. So the fifth one is called centromere. Centromere is also, we find antibodies against centromere. Of course, remember what is centromere. Centromere is a part of chromatin or chromosome, right? So anti-centromere antibodies react with the centromeric chromatin of metaphase and interphase cells. Those cells at the stage of interphase and metaphase, they suffer anti-centromere's antibody reaction. So the particular pattern on tissue culture cell is discrete or speckled. Sometimes centromere reaction is not a pattern, but those are the antibodies against centromere. So when they are reacting, they react as discrete or speckled reaction. So these antibodies appear to be highly selective for CREST syndrome or CREST variants of PSS. You remember what is called CREST syndrome is a variant is a variant of systemic sclerosis or progressive systemic sclerosis, which is characterized by calcinosis, Raynaud phenomena, esophageal motility abnormalities, sclerodactyl, as well as telangiectasia. So I will explain these terms later. What is calcinosis, Raynaud phenomena? This one we have seen it. Esophageal motility abnormalities, as well as sclerodactyl and telangiectasia. So these antibodies is found infrequently in serum of patients with the systemic sclerosis. I mean SLE, MCTD, and PSS. So this is antibody against centromere. Antibody against centromere, it is found in PSS, but not in all types of PSS, but it is PSS with the CREST variant. So, or CREST type of PSS, but it is not frequently seen in other conditions. So that's why when you are asked what is anti-centromere antibody, it is antibody which is normally detected in PSS of CREST variant, right? So then it will just react with the anti-centromere or centromere of the chromatin or chromosome. So, and in the interphase and metaphase of the cell division, but it can react as discrete or speckled. So centromere is not a pattern. Centromere is just a part or antigen. Then anti-centromere antibodies will react as speckled reaction like this. It is not really it is not really a full speckled, as you can see, but it is a bit speckled. It's not a full one, so you can see there's many gaps here, which is not really a full non-speckled reaction. This is what we call anti-centromere antibodies reaction. So I was trying to explain these terms, the terms we discussed in CREST syndrome. We have telangiectasia, that is of course, which is thread-like red lines of pattern of venules of the skin, and the sclerodactyl, it is tightening and thickening of the skin of the finger. That's what you call sclerodactyl. Sometimes this is of course a syndrome or a manifestation. Yes, please, I will just take your question after we finish this one paragraph here. Sorry for that. So, and Raynaud phenomena is a condition that causes the blood vessels in extremities to narrow and restricting blood flow, and usually occurs in the response to stress or cold exposure. So these are some of the terms. I just try to explain them because sometimes they would look a bit unusual terms in this immunology course. So finally, we have also another technique that we use to detect those antinuclear antibodies, but not commonly used. It is rapid slide test for antinuclear proteins. For example, we have SLE latex test, which provides a suspension of polystyrene latex particle coated with the DNP. So this is just only specific for deoxyribonucleic protein. It is just a rapid test for that. And of course, remember that DNP is associated with the SLE and also Sjogren rheumatoid arthritis and scleroderma. You remember that anti-deoxyribonucleic protein is associated with this condition, SLE, and remember that DNP is nucleoli, right? Anti-DNP are associated with nucleoli, nucleolar reaction, right? So that is what you find most commonly, of course, in SLE and also rheumatoid arthritis, scleroderma, and Sjogren syndrome. So also, lastly, we have also sometimes we can use autoimmune enzyme immunoassays in order to provide qualitative screening tests for the presence of antinuclear antibodies. So in one well, the assay collectively detects total antinuclear antibodies against the double-strand DNA, antibodies against histones, SSA or soluble substance A, soluble substance B, Smith, SM / ribonucleoproteins, Scaro70, GO1, anti-centromeric antigens. All of them can be detected using autoimmune enzyme immunoassays, but it is also not commonly used. So what we use commonly here, it is fluorescence technique to detect antibodies against nucleus or nuclear components. Thank you so much for your time. So I'm going to receive a question.